You all not talking this morning, huh?
Yes, you're right. Week 11.
Oh, there you go. Talk with you, huh?
Yeah.
So you look at this week is actually um, it's full of lecture material. We got a few quiz questionnaires behind it, but we don't have label and coloring at this point. So part of this week, I want you to make sure you spend time doing that research. Most of these first two topics are quite, well, I shouldn't say straightforward, but they're fairly linear in integration. The endocrine system, let's just go to that real brief and touch on that. Um, I gotta come up with a better way of doing this small and big thing here. Endocrine system is over here. I prepared. All right. So the endocrine system is the first one that we're going to touch on. And basically, that system deals with hormones, which are small little chemicals that the body produces and then ships through the bloodstream, and they go to different organs, so-called target organs, and then on those organs they got an effect, a physiological change and effect. For example, the good one that we often use is the insulin, the blood sugar. It's a big, isn't it? And the blood sugar one, the insulin, is the hormone that the pancreas secretes. Here's the pancreas. When the blood sugar gets high, when we eat a lot of sugary things and we have it in the blood and we want to get it out of the blood because it makes the blood sticky, and that's not really that great. And so the insulin gets secreted into the bloodstream and goes to the body cells that are pretty much all of the body cells, and tells the body cells to pick up oxygen from the bloodstream to extract it, and then of course use it in their cells for energy. Glucose is the main use for energy. And then once the blood sugar is in the cell, the insulin will detach from the cell, from the receptor, which is right on the cell, right on that target, and then the door will close and the insulin will not go in anymore. I mean the glucose. In case we don't have enough blood sugar in the blood, we have another hormone also secreted by the pancreas called glucagon, and that hormone then goes to the body cells and tells the body cells, attaches to a receptor, that's then the target cell, and tells that cell to get rid of some oxygen. I mean, some actually tells mostly the liver to put some glucose into the bloodstream to raise up the blood sugar so we can function. For example, we get not enough blood sugar for the brain, so we get tired. The brain doesn't really store that much blood sugar, the brain cells. And so that's kind of what the hormones do. The hormones are chemicals that are secreted at a gland and then going through the bloodstream and attach to a target, to a receptor at the place where we need an action happen, and then the target cell does the action. So the hormone is a messenger. It's not really doing. It's a message telling the cells what to do. It doesn't do it itself. The cell does all the work. So this is all kind of described in here. The endocrine system works very closely to the nervous system. So what we just described is right here in this section over here. When we say the hormone gets secreted at a gland and then goes to the target via the bloodstream, and the target is where the receptor is and where the action happens in the nervous system, the same thing happens or many similar things happen. You got a messaging system, but this one, the chemical gets secreted in the nerve cell itself, in the what the gland was, and then the nerve cell itself has a extension. This time it's not the bloodstream, and that the chemical, in this case it's called a neurotransmitter, not a hormone. The chemical goes all the way down the pathway, it's called the nerve itself, the pathway to the end organ where it has to go to, and then at that point there, it releases the chemical to the target organ. And in the nervous system, the target organ is always going to be either a other nerve cell, like when we think, we have to have nerves talk to nerves, or muscle cell when we contract muscles. That's directly done with the nervous system, or a gland, and then the gland does the hormone thing after it's stimulated by the nerve cell. You can see that, for example, um, well, adrenaline is sort of in between. We'll get to that later. Um, but you see long-term effects on that when we look at the psychosomatic kind of stuff. But that then when we further evaluate these two systems, the thing is the nervous system is a very rapid control system, so we need to use that system, for example, for you're going down the freeway and you need to swerve out of somebody who was almost going to hit you. That fast reaction is the nervous system. The endocrine system is much slower, and you can notice that, for example, in terms of its speed, like after you swerved out from the jerk who almost cut you off, like a half minute later or so, you start getting the jitters if it was a real bad one. And the jitters is the adrenaline, and the adrenaline is actually what I was talking first, the nerves that goes to your adrenal gland, which sits behind your back on the kidneys, and then that releases the adrenaline, which is a hormone, into the bloodstream, and that hormone slowly has to travel down the bloodstream into the target cells, and that's when you feel that 15 to 30 seconds delay from the event to the shaky kind of feeling. That's that distance of time that it takes to get to the target cell. So it's not really that long, but it's long in terms of body stuff. If it were to take you 15 seconds to swerve out of the that would be too late. That wouldn't work out. So that's kind of the difference between the two of them. And then we also, when we look at hormones, we differentiate. As you probably noticed already, a lot of things are differentiated in the body between hydrophilic or hydrophobic, water loving or water fearing or fat loving. And that often then has to do with how things travel through the body. And generally speaking, the hydrophobic, or look at this, the other word here is lipophilic. Philic means loving, and lipo is lipids. So you can say something that rejects water is hydrophobic, but you can also say at the same time that substance, since we only have water and oil, will be also then lipophilic because it likes the lipids. It hates the water, it likes the lipid, the fat. And so those substances, the affinity to fat substances, they can penetrate and travel freely through a cell membrane. Just like here, so in here. These are called steroid hormones in the body. When you see steroid hormones, you know it's fat. When you see peptide hormone names, if you see something like peptide so-and-so, you know it's water-loving, it's hydrophilic. The problem with, well, it's not a problem, it's just a difference. The difference with the hydrophilic hormones, the ones that like the water, they can penetrate an oil film. And if you remember, a cell membrane is really an oil film with the hydrophilic on top and the hydrophobic in the middle. And so the peptide cannot penetrate that oil film. It has to stop and has to relay a message to the inside via what we call here a second messenger system. So that's you're going to get through that more into the test. I just want to touch on it real quick. And then the last one I kind of wanted to talk about for this chapter is the different hormone systems. So we have one system of hormones that is like the insulin and glucagon that I explained before. Like the blood sugar goes up, we need a hormone that brings the blood sugar down. The blood sugar goes too low, look at this one. We need a hormone that brings the blood sugar back up. And so that's a negative feedback time type situation. And a lot of the hormones are ultimately measured in a negative feedback type measuring system because that makes sense. You have too much of it, you need to slow it down a little bit, tone down the temperature. You have too little, you need to bring it up a little bit. So that's what that is. The question then just arises what's the control of that? So often what we have in this system, because hormone stuff is very delicate, that's why I would like people to think a little bit about Ozempic because it is a hormone. And we don't know long term when we must mess with hormones what that does. I mean, that's my only caveat on that. If interested in that drug, make sure you think about, or make sure you look at that a little bit. I haven't researched it myself. I don't know. It's the same with melatonin. You got to be careful not to take it too much. But often the negative feedback loop happens right at the level of the gland itself, like the pancreas measures the sugar stuff. It's a chemoreceptor here. But we often also have receptors in the brain, in the hypothalamus. And so the hypothalamus becomes a very important organ or part of the brain that controls our hormonal system because we measure different substances there. And so then that part of the nervous system communicates with the pituitary gland, which is sitting here in the sella turcica, which we've learned in the brain, in the skull. The reason why we have that sella turcica there is because a nervous tissue is very slow growing, glandular tissue is very fast growing. And so the gland, we need to make sure we encase it in bone so it doesn't grow into the brain and destroy the brain. So sometimes when that happens, then people have vision disturbances because the vision pathway goes right above here. And so then the hypothalamus talks to the pituitary gland and tells the pituitary gland, which is right here, should we make more hormones or should we inhibit more hormones, make less hormones here in the pituitary gland? And look at that. The pituitary gland releases all these different hormones. Here's a labeling thing of what they all are. We go through all the details. The thing is, when you go through these, it says like, well, the adrenocorticotropic hormone does this, and then the thyroid stimulating hormone does this. And so it's fairly linear. And you kind of have sort of table, you make it tables of what does. And, we talk about here, let's see here, where is that? For example, where is that? These are the ones I don't want to talk about. Like the adrenocorticotropic hormone, for example, is the one that's simple to understand it. So then that one goes to the adrenal gland and releases the hormone at the adrenal gland. So you have a multi-step process here to ensure control of how much hormone gets released into the bloodstream. So what I want you to do is go through all these different hormones and read through them, what they do, and then we got this quiz thing about it, or these questions about it. You go through those too, and then slowly the data comes into the brain. The other thing, of course, make sure you go to the test review and have that next to you when you study. That way you know exactly what I want from you as knowledge. Then there is also a couple hormones that the brain secretes themselves, and this is here in green. You'll go through those. It's not that complicated, that part. And then, the pituitary gland is the one that secretes a lot of these hormones into the body. And then let's see what else we got that I need to touch on in this chapter. This is all stuff you can read through. It's in the videos. The thyroid gland, the bone growth, calcium. Calcium is very important. Calcium is like blood sugar. It's like we need calcium in the blood all the time. If we don't have calcium, just remember the muscle contraction. What would happen to the muscle contraction if you don't have calcium in the system? It wouldn't be good. And then we have the adrenal gland. It's an interesting gland. It has multiple areas that secrete multiple different hormones. And again, I don't need you to memorize these hormones, right? I need you to understand what these things do, and then we can apply them in the quiz and then in the test. But you have always these different reviews. Let me make sure you guys got the review. You remember where the review is, everybody?
You said where it's at?
Yeah.
Yeah.
In the back.
It's in modules?
It's in that module. Let me show you real quick. Isn't that down here? Ooh, no, not that far down. Test 3, test review. Right here. You click here. Hopefully it doesn't take forever. This comes up and you can print it out or just download it or copy it, or it doesn't matter what you do. But these are the things I want you to know for the pituitary gland. I mean, for the endocrine system. These bullets right here. you need to take a screenshot, do it right now. I know that might be a little overwhelming at first, but I think once you go through it, it should be fairly straightforward. The again, if you have questions, butt in. Don't wait for me, okay? The next topic that we're gonna talk on is in the skin and the integumentary system. And when we look at the integumentary system, that one is even more straightforward. I mean, I shouldn't say that because it's not all that easy, but compared to some of the things like we had, the immune system is pretty intense. I don't know. How did you guys like the immune system?
I liked it.
Good. Yeah, it's
It was
certainly
interesting.
it's an interesting topic, that's for sure. It can get a little elaborate depending on how deep we go on it. Okay, then when we do the skin, let me go to the skin myself real quick. Do you guys see the skin?
Yes.
All right, good. So the skin is actually, I'm just going to stay on this for the skin because we really talk about a top layer skin called the epidermis. We're going to talk about different layers from bottom to top because the skin is kind of interesting. You got a dermis, a base layer, and underneath. If you cut yourself and you bleed, you hit the dermis because the epidermis does not have any blood supply because the epidermis on top is kind of our leather. It's sort of cells that grow and then slowly die off, and then they fall off as flakes. And so it's kind of an interesting system. I mean, it's a little gross, but it's really cool because when you hit yourself somewhere, you don't bleed. It's just a few cells fall off, and then they keep growing from underneath. And so when we look at the epidermis, these layers here, these describe that what is really is a creation, the basales, where we make the cells and then we accumulate a certain protein called keratin, and that's the leather protein, the waterproofing and hardening protein. I should call that the leather protein now, because that's really what that does. And then the basically after that, the cells dry out, and at some point they become scales, and this is the corneum, is the scales. And then in the dermal layer, the interesting part there is everything from feeding blood supply nutrients to also nerve energy through the epidermis comes to the dermis. We don't have anything blood supply or nerve supply in the epidermis itself. And the top layer, it has two layers. The top layer of the dermis is also interesting because it has a lot of hills on it. And so what that does, they go up and down and yeah, create little hills, papillas, they call that. And that increases the surface area dramatically, and that makes the skin really be taut and hold on to the underneath skin, the dermis. The epidermis holds on to the dermis. And then we talk about a few cells. Main cell is keratinocyte. Keratin is the leather protein, so most cells are making leather proteins. And then we got a few nerve ending cells, deep pressure, superficial pressure of Merkel cells. And then we also have skin color cells. The melanocytes are the ones who make the skin color, and they're basically a protection for UV light. What that is, and it's kind of cool because it's little cells that make umbrellas to protect the basic layer of cells, the ones that regenerate all the time, from the damaging UV rays, and so that creates that darker color in our skin. Stuff to think about. And then Langerhans cells, those are cool because that kind of means long johns. Langerhans is a German word, and it means long john. And those are actually the immune cells in the skin itself. So we have macrophages. When we remember the immune cells we learned in the blood, the macrophages are the big eaters, and they often, I think I said it there, they squeeze into the body tissues and become resident macrophages, and those are then having different names, because when the people studied the skin, they didn't know that those were blood cells at first. They just saw different things and they gave them different names. And so the long john cells are the macrophages that reside in the skin and keep our skin proper and not infected. And then a little function of all of that. What does it do? Look, it protects us, obviously. we know now the immunity, make a boundary is a great immune thing. And then it helps us for not losing water. Essentially, it's like, we came out of the ocean, so to speak. And so we have an ocean inside and a skin suit around it. So that prevents water from getting out. Of course, it's not as simple. But what's interesting is the salinity of our system is fairly similar to the ocean, how much salt our system has. And then it, of course, has sensory function. You could touch, you could feel, you have temperature control. And interestingly, if you think of a face, not the expression as much, but the blushing, for example, or the white coming in, it's communication. It's a communication tool as well. Non-verbal, of course. And then finishing up the chapter is appendages, and those are the glands, the sweat glands and the oil glands in the skin. Those are the ones we have. And each hair that we get, which is here too, an appendage, is actually associated with an oil gland, a sebaceous gland. We'll get to that. And then last topic will be the nails. That's, of course, a part of the skin as well, which is kind of helpful because without nails, could you imagine pinching something and picking something up that needs a backstop, a hard back to pick up? That might be quite difficult. So yeah, it makes us think a little bit different about these structures when we look at the functionality of it. All right, that's pretty much the skin. Any questions to that?
No.
That's pretty good, huh? And then the next topic will be, and this is, we finish up with the beginning of the nerves. I know, I'm sorry, I'm just kind of blabbing here. If you have questions, just butt in or comments too. This is too boring. All right, nervous system. Let me get it on my end. So now we're getting into this stuff, and I love this stuff. So the nervous system, we'll be talking about that for a little time now. It's electricity, how it starts. And electricity is based on a few concepts. And so we need to get that understood. And so I want to just give you a very brief general overview of that. When you look at electricity is an exchange of an ion. And so it's electrons moving creates that electric shock thing. And so it's charges, it's positive and negative charges moving across a boundary. And so that's electricity. And so now when we look at the system, the body, we don't really, we have to figure out how to make electricity, or I guess the Lord did that. But we can use ions because ions are charged molecules. And so we can take ions, these sodium and potassium and calcium and all these charged molecules, the ones with a negative or a positive on top, because that indicates their charge. And so we can transfer those from one side of a cell membrane, which is a boundary, to another side of a cell membrane, and that exchange of a charge is going to give us electricity. Does that make sense?
Yes.
Good. So far, so good. And the next problem, though, is now that we need to understand is we want to create electricity, which is done by the charge moving across the membrane. See, you can see that here. The charges move here. This is the charge move. But who's carrying the charge is a chemical. And so now we need to move the chemical in order to create a movement for the charge. And so that's then when they start talking about an electrochemical event. So that's where it gets a little complex, because we need to concern ourselves now first with the chemicals to then be able to move the charge. And so the chemicals that we use in a nerve impulse, you guys understand, right? Opposites attract. A negative will attract to a positive, right? That stuff is okay?
Yeah.
All right, the magnet stuff. And the boundary we just talked about. And so now the problem is, if we want to move these chemicals across cell membrane, this is cell membrane, these are all the chemicals. See, then you have pluses, these are the charges. So in this situation, what we've done, we've created a situation where all these sodiums are on the outside, right on the outside of a cell membrane. And then we put all these potassiums all on the inside of the cell membrane. And we do that with a physical pump that continuously pumps sodium out and potassium in. And so now we create this chemical environment where chemically speaking, we have a huge concentration of sodium on the outside. Where do we see that here? We have a picture that shows that better. Nah, I guess not in this slide. And we have a small amount of sodium on the inside. A huge chemical gradient. Remember chapter 2 when we used to talk about the movement of substances across the cell membrane? How the chemical gradient is the factor that makes something move passive versus active. Do you remember that?
Yes.
Good. So, because the cool thing is, a passive movement across the cell membrane, we can create much faster than an active one. You think about that. If you're up on top of Mary's College, and you bicycle down, goes pretty fast and easy compared to going uphill. It's the same kind of concept. And so the pump is what uses energy, and that takes much longer. So in a peaceful times when we don't have a nerve impulse, what we're going to do is we're going to put all these chemicals to the outside and all these potassiums, the K stand for potassium, to the inside of the cell membrane. And then when we have a nerve impulse happening, this is kind of what we're working towards. Give me a nerve impulse. When we have a nerve impulse happening, all of a sudden, we do this. All of a sudden, we open gates, channels that only let sodium through. And we have a huge amount of sodium. This is not indicated here, but we have a huge amount of sodium concentration right on the outside of the cell membrane and hardly any on the inside. So all of a sudden, when we open these doors, this sodium wants to go to the inside of the cell. What does it do when it does that? It brings in all those charges with it. So it changes the charge on the inside and make it much, much more positive because it carries all these positive things to the inside. And that shift of bringing that positive in, that is a nerve impulse. So we change the electricity, the charge level of charge on the inside. Actually, what they call that voltage. When you change the difference between a charge on the inside and on the outside. And so when we actually measure that difference, that voltage in a normal state without a nerve impulse, it says it's 70 millivolt more negative on the inside as compared to the outside. To us, that's just a number. But what happens when we bring then all these positives to the inside? That negative 70 on the inside that is measured is no longer negative 70 millivolts. It goes towards zero. It gets less because we bring positive charges to the inside. And when we reach, well, we're going to reach about positive 30, bringing all these sodiums in. And that's the peak of the nerve impulse. That's when you hit your funny bone and that nerve fires to the pinky. Have you ever done that? Hit the funny bone? That's it, man. That's a nerve impulse. That's what happens when you do that. And that hit stimulates those sodiums to go in and say, like, well, something's weird. So a pressure on a nerve can stimulate some of that too. And so once we have reached that peak of a 30 millivolts positive on the inside, what are we going to do? We got to have to do something with that too much positivity. So we have to bring the positive energy back to the outside. Now, we can't do that with the sodium anymore because we already brought it all in and now it's balanced. Remember in chapter 3, the chemicals want to be balanced across the membrane if we don't have a boundary. And so now we can't use sodium anymore to do this, take away this firing nerve impulse. What we have to do now, we have to open doors for potassium. And in the other slide, the potassium is a huge concentration on the inside of the cell and not much on the outside of the cell. And so guess what? Now we're going to take all that positive energy, these charges, and throw them back out of the cell. And from this positive 30, we go all the way back to negative 70. And then that's the finish of the nerve impulse. Still good?
Yeah. Yes.
That's good. And then the only thing left is that we got to reshuffle the chemicals to make sure we put all the sodium back out and all the potassium back in. And at that point, when that happens, you can fire another nerve impulse. So all of that takes, of course, milliseconds. It doesn't take long, right? Because sometimes the nervous system is interesting when you need to fire a stronger impulse, like when a stimulus is really strong. You don't do that for making the nerve impulse stronger because a nerve impulse is always the same strength. What you end up doing is you end up rapid firing one after the other. And so that increases the strength of an impulse. So this stuff takes care in very fast, rapid order. And the restoration of the chemical environment is done by the pump. That's the one up here, wherever that was. This one, that pump. That's what creates that gradient. Okay, well, if you've gotten that far, everything else is peanuts because this is the big kahuna for this chapter, this nerve impulse, and we call the nerve impulse also an action potential. And this is here where we talk about how the action potential it's an all or nothing event. That means if it happens, it just happens. You can't stop it. It's like a bullet that gets fired. Once it leaves the barrel, you can't stop. But what we do is when we want to have a more intensity of an impulse, we fire rapidly after one another, and then that increases the intensity of the impulse. So that just shows that. Anything else is, oh, this is kind of cool to finish up maybe on this chapter. So this one is called how do we propagate the nerve impulse? How do we make it fast go down the long nerve fiber, the nerve itself, to where we need to go? How that firing happens so rapidly, and what the body does, it has helper cells that create an oil film, and they wrap that oil film, sort of a plastic wrap kind of thing, around that long extension that the nerve impulse needs to fire down, the nerve itself. And it leaves little gaps at various spaces. And then the nerve impulse the chemicals can exchange where the wrapping is around, the fat layer. So what they end up having to do is they have to jump from one place to the next place. And so they have to draw, the opening of the channels from here has to be drawn down and it has to be felt that far down. And then that speeds up the impulse much faster instead of just trickling down little by little. So that's kind of cool. But the cool thing there is look at that. Saltatory conduction is that called. So they like the jumping is the saltatory. Anyway, once you get through this chapter, it will probably make a little bit more sense, this part. And then lastly, I think, let me pull up, where am I here? The anatomy thing, and we can finish up with that. Here we go. Oops. What am I doing? There we go. And stop it. Thank you. And in terms of neuroanatomy, we make this a little smaller. There we go. We'll do the brain next week. So this is the small anatomy stuff, not the big anatomy stuff. So we have in the nervous tissue, we have nerve cells, the ones that do what we said they would do, the nerve impulse, that what we just described. And then we also have glial cells or neuroglial cells, and glia means glue, and they help support the nervous system and provide nutrients or do that insulation like we just talked about with that saltatory conduction. That's also a job of a neuroglia. We don't really touch too much on the specifics of them. We go mostly into the neuron in this class because that's really the one who does the job that we're looking for from the nervous system. So when we look at the neuron, they are functionally and structurally independent units. So they function by themselves, and they connect to other neurons, to muscles, or to glands by what we call synapses. These are synapses here. I said before, they only connect to neurons, to muscles, and to glands, to nothing else. That's kind of been helpful for me to understand that system a little bit more. When we look at the structure of a neuron, a generic neuron, we have neurons that look different. We have neurons that look like that. We have neurons that look like that. We have neurons that look like that. And then we even have other neurons. Where are those? Well, we'll get to those. But they all generally have the same parts. They have receiving extensions. These are called dendrites. Then they have a cell body. And then they also, and that's where we do, I gotta close this window real quick. I'm sorry, somebody's being loud outside. I am not able to focus. Children playing. And then we have the cell body, and that's where we have all the metabolic stuff, where we make the neurotransmitter, the chemicals, where the energy is made, etc. All the little organelles that we talked about in the cell chapter. And then we got one long extension, which is what we collectively then call the nerve. Like the funny bone here, that's the nerve that we hit. That's made up. Actually, it's made up of both dendrites and axons, but for our purpose right now, the big one there is the axon, which shoots the nerve impulse down from the cell body to the axon terminal, which is where we have the connection then to the next nerve or muscle or gland. And so that's how that looks. Oh, see here, this is another one that looks like that. This is a weird looking, because when we classify neurons, we have some that have many extensions in the body and then one axon. We have some that have only one extension and a cell body in the middle. We only find those in a few places, in the nose and in the eye. Very rare. But then we have another set, which is kind of interesting. We have one long extension going from the dendrites to the axons, and it's basically kind of one long axon. And then the cell body is sort of tucked to the side, away from it. And what those nerves are, those are the ones that bring in information from the outside of the body to the brain, because the peripheral area, away from the well, let me talk about that real quick. Where was that? Oh, that was in the other chapter. Oops. Let me go back real quick. I'm very sorry about that. Where was that? Here we go. That was the first slide in there, I think. Yeah, here. We often, when we talk about the nervous system, what we think about is the brain and maybe the spinal cord, and we call that the central nervous system. We call that central because it's in the center, but it's the decision making process part. It's the brain and the spinal cord. It's like a computer that makes the decisions, and then everything else that comes out to the side, down the arm, down the legs, down the torso, around the torso, that's all just cables. Those are all just these cables where we send a nerve impulse down. And so that's called the peripheral nervous system, because it's not in the center, it's out in the periphery. It's like in the appendages almost, but away from the central part of the body. Okay, sorry about that. I did not mention that before. Now we can go back to this. So the ones that only have one extension in the cell body tucked to the side, those are the ones that carry all the information from the periphery to the central nervous system. And the problem is, in the periphery, that's dangerous out here. That's not encased in bone, my arm and my legs. That's muscle, and so a cell body is very delicate and can be damaged in an environment like that. And so what we do is we put them collectively. We are stronger in numbers and greater in strength when we have multiples, and so we put them collectively all along the spine, inside of the spinal bone almost, in some area where we then later on call the dorsal root ganglion. We'll see that the 2 chapters down. And so those are very specifically interesting looking creatures. compared to the other ones. They're only used in that area, and they're only used because the cell body needs to be protected, enclosed in somewhat in bone, but also with other cell bodies together, so they're stronger because they're a collection. So those are just the different types as we go through this chapter. And then the last thing I think importantly here, I hope it's the last thing. Yeah, pretty much for us right now is the synapse. And the synapse is where one nerve ends and we get the next nerve happening, or the muscle or the gland. And so that's what happens in the synapse. And so when you look at that, we have a receiving ascending end. We call that a presynaptic neuron first, and then we have a membrane, and that's then called the membrane. And then the interesting part is the nerve impulse comes down to this area here, the synaptic terminal or the synaptic knob, sometimes they call that. And the chemicals, that neurotransmitters, are already they're already here. They're being brought down from the cell body to this area over time. And then when the nerve impulse arises, all we're going to do is we're going to secrete those chemicals into a space, and then they float in. They have a gap. They have to float to a receptor on a postsynaptic organ, a nerve, a gland, or a muscle, and then they have a reaction there. And so we call that the postsynaptic membrane, where those receptors are located on. So that's kind of a cool part of that. So let me make sure we covered all of that. Yeah, I think the next parts we can pretty much go um, with the video, because that talks a little bit more about the receptors. And then this is kind of funny how you can have different connections. You can have a connection from an axon to a dendrite. They call that axodendritic synapse. But you can have one that goes directly to a cell body. They call that axosomatic, or an axonic if one goes to the another axon. And this one is actually kind of interesting because this one is involved in neuroplasticity and learning. And maybe I finish up with that. Neuroplasticity is the fundamental of learning, because when we fire a pathway, neurons together, over time they wire together. They get stronger and stronger and stronger. So that's true with a thought. That's true with a topic you're studying about, with learning about. Um, um, and how it does is the last neuron will grow extensions to the previous neuron, and it becomes sort of its own circuit. They call that a reverberating circuit, and it strengthens that thought. So the more you use that thought, the more it strengthens it. And that is why flashcards are really helpful and really important, because if we study something from beginning again in a card, let's say we test ourselves, and we have to look for the answer, we're not quite sure, but we find it in our head and we have it right. That stimulates these growths tremendously. And that searching, that kind of a little bit of pain, having to find for an answer, that is what makes those nerves grow. And I think the pain is probably when they actually grow a little bit. Who knows? Good. So that's kind of to me, that's actually the coolest part of the whole thing, because that's really applying it later. And we will have an exercise activity that we focus on gratitude, and that has to do with this. Anyway, so I think that's probably good enough from my end. How are you guys doing?
Doing all right.
Yeah, was okay information wise? Can you take it from here?
Yeah.
That was pretty good.
Good. All right, then you guys do that, and then we can come back together next week if you have any questions, or you just shoot me a text. I can answer questions too. Especially as it comes to the fundamentals of the nervous system. Just make sure you understand that, or ask me if it's a little tricky. But hopefully, the way we explain it today sets the foundation well.
I have a quick question.
Yeah.
I'm not sure if you answered this today. When do we come back to the lab?
I didn't answer that. I think the next lab is the 29th.
29th. Okay.
Let me double check. Yeah, we have Yeah, I know the first we had, but these holidays make me crazy. Well, they shouldn't because they're fun. Um, Let me see here in the
It's
syllabus.
just hard to get back into things.
I know, totally. Course outline. Yeah, 29. On the
Perfect.
29th,
Thank
we
you.
do the brain and the eyeball. So we're going through the brain and the eyeball in this next couple weeks, and then we're gonna go dissect those. And then we have one more scheduled on the 13th, where we dissect the kidneys, and then we'll also be doing the dissection of the heart in that class. Catch up with the first of April.
Okay, thank you.
So that'll be kind of fun. Yeah, if you like doing cutting stuff, come on in those days. That's what we're gonna be doing. And of course, we still have the cadaver labs available as well.